Hello all, welcome to Mediverse Atlas. So today our topic of discussion is erythropoiesis. So erythropoiesis is an important LAQ in the blood physiology. Okay, let's get into the topic. So before understanding what is erythropoiesis, we'll understand what is hemopoiesis first of all. Okay, as we all know that the blood contains various type of cells, right? Various type of cells like RBCs, WBCs, and platelets, right? So the synthesis of all these type of blood cells is known as hemopoiesis. Okay, so hemopoiesis is defined as the process of origin, development, and maturation of all blood cells. Okay, so now let us try to understand the definition of erythropoiesis. Okay, so the erythropoiesis is the process of origin, development, and maturation of erythrocytes. Now, what are these erythrocytes? These erythrocytes are simply nothing but the RBCs. Okay, so in short, the synthesis of RBCs is known as erythropoiesis. Okay, now erythropoiesis occurs at different sites and different stages in our lifetime. Okay, so there are three main particular stages of erythropoiesis. There are three main stages. The first one is the mesoblastic stage, the second one is the hepatic stage, and the third one is the medullary stage. Okay, so first we'll be discussing about the mesoblastic stage. This mesoblastic stage occurs during the intrauterine life, IUL, okay, in the intrauterine life. And we all know that the yolk sac, the yolk sac has three germ layers. We are reading this from our lower classes, right? So there are three germ layers, okay, ectoderm, endoderm, and mesoderm. So, in the mesoblastic stage, as the name suggests, meso, so the erythropoiesis, that is the synthesis of RBCs, takes place in the mesoderm of yolk sac. Okay, mesoderm of yolk sac. I'll just highlight it. The erythropoiesis takes place in the mesoderm of yolk sac. And the erythropoiesis which occurs in the mesoblastic stage is intravascular. Okay, so what are the points to be remembered in the mesoblastic stage? It occurs during intrauterine life. Erythropoiesis takes place in the mesoderm of yolk sac, and erythropoiesis is intravascular. Okay, now let's try to understand what is hepatic stage. As the name suggests, hepatic. What does hepatic mean? Hepatic means liver. Okay, so this hepatic stage starts from the fifth week of gestation. From the fifth week of gestation, and the erythropoiesis, that is the synthesis of RBCs, takes place in the liver and spleen. Okay, uh, in the liver and spleen at the fifth week of gestation, the erythropoiesis takes place. This is hepatic stage. Okay, now we'll understand medullary stage. First of all, what is this medulla? We are talking about medullary stage, right? So what is this medulla? We all know that the bone, when we look at the structure of bone or the anatomy of bone, it has medulla inside the medullary cavity. So this is the medulla inside the bone. So the medulla, in the medulla, erythropoiesis takes place. That is the medullary stage. Now this medullary stage has different, again it has some different types. Okay, that is from the fifth month of IUL, from the fifth month of intrauterine life to adolescence or up to 20 years. Again from up of adolescence, that is up from 20 years to lifetime. Okay, so from the fifth month of intrauterine life to adolescence, the erythropoiesis takes place in the medulla of all long bones, medulla of all bones basically. Okay, but after 20 years, the erythropoiesis is limited to only certain bones such as sternum, ribs, iliac bones, vertebra. Okay, so erythropoiesis is limited only to certain bones after the adolescence or after 20 years. Okay, see, please don't confuse here. Hepatic stage is at the fifth week of gestation, whereas the medullary stage at the fifth month of gestation. Okay, please have a clarity. Hepatic is at the fifth week, medullary is at the fifth month. Okay, see, medullary stage is from the fifth month of intrauterine life till adolescence. Erythropoiesis is done in almost all the bones of the body, long bones and flat bones. Okay, almost all the bones, where the medulla of the long bones or medulla of the bones, right? After the age of 20, erythropoiesis is limited to membranous bones like vertebra, sternum, ribs, scapula, iliac bones, and from the ends of long bones. I hope you got the point here. Okay, please try to understand. At the fifth month of IUL to the adolescence, erythropoiesis is done in almost all the bones of the body. But after the age of 20 years, erythropoiesis is limited to only certain bones such as vertebra, sternum, ribs, scapula, iliac bones, and ends of long bones. I hope you got my point. Now, we'll just discuss a small clinical point here. See, the medullary erythropoiesis, right? From the up to the age of 20, medullary erythropoiesis takes place. Now, this extramedullary erythropoiesis in the postnatal life is always considered abnormal. What is this extramedullary erythropoiesis? Extramedullary erythropoiesis is nothing but the synthesis of RBCs out of the medulla. I mean, at the liver, at the spleen, or at the different sites of the body, except at the medulla. Okay, synthesis of RBCs outside of the medullary cavity after birth is abnormal. Okay, why it is abnormal? Synthesis of RBCs is good only na. Why it is abnormal? Because this uh, it indicates the bone marrow dysfunction. We all know that the medulla of the bone contains bone marrow. In the bone marrow only the RBCs are synthesized. Okay, so now extramedullary erythropoiesis, that is the outside of the medulla of the bone, at the liver or at the spleen. If the erythropoiesis takes place, it is considered as abnormal. Why does it happen like that? Why extramedullary erythropoiesis happens is due to the bone marrow dysfunction. Okay, if there is any bone marrow dysfunction, there will be extramedullary erythropoiesis. If there is extramedullary erythropoiesis, the RBCs are synthesized in the liver and spleen. Due to excessive synthesis of RBCs in the liver and spleen, it leads to enlargement of liver and spleen, which is known as hepatosplenomegaly. Okay, so hepatosplenomegaly can take place when there is extramedullary erythropoiesis. It is abnormal. Okay, now here we have one more note point. See, after 20 years, the shaft of long bones contains yellow bone marrow due to fat deposition and loses the erythropoietic function. Okay, so this is the reason main erythropoiesis is limited to only certain bones after the age of 20. So what happens is the bone marrow which is present in the long bones, due to the accumulation of more fat as the age prolongs, it converts into the yellow bone marrow. When it is converted to yellow bone marrow, the erythropoietic function of this bone marrow is decreased. So only the erythropoiesis is limited to only some bones in the or of the after the 20 years. Okay, I I hope you got my point. Okay, now let us discuss the main topic which we need to focus. It is the process of erythropoiesis. Okay, before discussing the process of erythropoiesis, we need to know what is this. What are these stem cells? We'll be hearing na stem cells, preservation of stem cells. So, what are these stem cells? The stem cells are nothing but the cells which are capable of self renewal and self-differentiating into specialized cells. Okay, so they do not require any other factors to get renovated or to get differentiated. They are capable of their self-renewal and self-differentiating. Okay, so if the stem cells are designed to synthesize the blood cells, they are known as hematopoietic stem cells. I hope you got my point. Okay, what are the hematopoietic stem cells? The specialized stem cells which are specialized to synthesize the RBCs. Okay, these are the hematopoietic stem cells. So these are primitive cells in bone marrow which give rise to blood cells. Okay, so now these hematopoietic stem cells are also called as uncommitted pluripotent hematopoietic stem cells. Uncommitted pluripotent hematopoietic stem cells. Now, what is the meaning of these uncommitted pluripotent hematopoietic stem cells? See, as the name suggests, uncommitted. What does uncommitted mean? that it is not committed to a particular thing, right? So uncommitted pluripotent hematopoietic stem cells are the stem cells which are capable of synthesizing all type of blood cells. Okay, uncommitted pluripotent hematopoietic stem cells are the stem cells which are capable of synthesizing all blood cells. Okay, whereas committed pluripotent hematopoietic stem cells are useful for the synthesis of particular type of blood cells. Okay, see, they synthesizes a particular type of blood cells. Which one? Committed plurihematopoietic stem cells. Whereas uncommitted plurihematopoietic stem cells are capable of synthesizing all type of blood cells. I hope you are clear with this committed and uncommitted pluripotent hematopoietic stem cells. Okay. Now we'll just have a brief overview about the erythropoiesis in this flowchart. I want you guys to remember this flowchart. Okay. See, we have discussed about uncommitted pluripotent hematopoietic stem cells or UPHSC. Okay. See, in the bone we have bone marrow. In the bone marrow, we have stem cells. Which stem cells? Hematopoietic stem cells. Okay. The hematopoietic stem cells are initially uncommitted. Okay. In the initial stages, they are uncommitted initially. They are later committed pluripotent. They are later converted into committed pluripotent. That is, this committed pluripotent are capable of synthesizing different, particular blood cells. Na? So, this committed pluripotent hematopoietic stem cells can synthesize colony forming blastocyte, which is all known as megaloblast. Okay, or lymphoid stem cells. You see, this is the thing which we need to remember. Okay, we have to remember this. Okay, so this committed pluripotent hematopoietic stem cells synthesizes colony forming blastocyte and lymphoid stem cells. Okay, now this colony forming blastocytes again forms different subunits. Okay, colony forming unit TM. CFUM. We call this like this CFUM, which synthesizes megakaryocyte, that is platelets. M for megakaryocyte. Okay, same colony forming unit E for erythrocyte. Okay, which synthesizes RBC, and colony forming site GM. GM means granulocyte and monocyte, which are capable of synthesizing granulocytes and monocyte. Water granulocytes, neutrophils, basophils, and eosinophils. And monocytes is a agranulocyte. Okay, and we have lymphoid stem cells, which synthesizes lymphocyte. Okay, so committed pluripotent hematopoietic stem cells synthesizes again two different type of stem cells. Okay, so that it synthesizes two different type of cells, that is colony forming blastocyte and lymphoid stem cell. Again, colony forming blastocyte or megaloblast synthesizes three different specialized cells, that is colony forming unit M, colony forming unit E, and colony forming unit GM. Okay, now this M, colony forming unit M synthesizes platelets, colony forming unit E synthesizes RBCs, colony forming unit GM synthesizes WBCs, most of the WBCs. Okay, I hope you are clear with this flow chart. Okay, now see, this is one of the pluripotent hematopoietic stem cells which convert into CUFE, right? CUFE. First, it is converted into colony forming blastocyte or megaloblast. Now, this is converted into CUFE, that is colony forming unit E, which symbolizes erythrocyte. Okay, colony forming unit E synthesizes erythrocyte. See, synthesizes erythrocyte. So, this colony forming unit E undergoes various steps to synthesize a matured RBC. We need to study all these steps in detail. Okay, uh, to know how an erythrocyte is formed. Okay, so the CUFE, the colony forming unit erythrocyte, undergoes different stages. What are the name of the stages? Proerythrocyte, early normoblast, intermediate normoblast, late normoblast, reticulocyte. So CUFE undergoes various type of stages. It crosses various stages to form a mature erythrocyte. Okay. Now during this process from the CUFE to erythrocytes, so what are the changes which are occurring in each type of cell is the first, the reduction of size of cell. Okay, first the size of the cell decreases. Okay, as we can all see in this image, see this is large as compared to this. So reduction of the size decreases. Okay, now disappearance of nucleoli and nucleus. Okay, see here nucleus is present, nucleus is there, nucleus is there. See, nucleus is disappearing slowly. Okay, it is slowly degenerating and disappearing. So this is also one of the important change. So there will be disappearance of nucleoli and nucleus. And next, appearance of hemoglobin. See, hemoglobin is red in color, no? See, a mature erythrocyte is red in color. So there will be synthesis of hemoglobin as as the CUFE passes through various stages. And there will be changing in staining properties of cytoplasm. Cytoplasm is stained differently at different stages. Sometimes it is stained as basophilic, sometimes it is stained as acidophilic. So there are different stages in which differently cytoplasm is stained. We'll be studying. So what are the different stages which we need to study? The first stage is the proerythroblast. It is also known as pronormoblast. And the second stage is the early normoblast. The third one is the intermediate normoblast. Fourth one is the late normoblast. Fifth one is the reticulocyte, and sixth one is the matured erythrocyte. Now, we'll be discussing each and every stage in detail. So, the first stage is the pronormoblast. Okay, now, what are the characteristic features of this pronormoblast? So, see, the nucleus, the nucleus and the size of the cell are large initially. Okay, and the nucleus occupies almost 80% of the cell volume in which proerythroblast. Okay, so in proerythroblast, nucleus is large and occupies 80% of the space of the cytoplasm, okay, in proerythroblast. And cytoplasm is basophilic due to the presence of high concentration of polyribosomes. As there is high concentration of polyribosomes, what are these polyribosomes? They are nothing but the ribosomal RNA. We'll be studying about this ribosomal RNA in detail in biochemistry. Okay, so till physiology, please remember, due to the high concentration of ribosomal RNA, the cytoplasm is basophilic, in which in pronormoblast. Okay, and the nucleoli are present. Okay, and the nucleoli are present in the nucleus, and mitosis is also present. Okay, and hemoglobin is not yet formed. As this is the first stage, na, hemoglobin will not at all form. Okay, so these are the characteristic features of pronormoblast. The first point is the cell is large. So the cell is large, nucleus is large, occupies 80%, nucleoli is present, mitosis is present, and HB is not there, in which in pronormoblast. Now we'll be discussing about early normoblast. Now, what is this early normoblast? It is the first erythroblast to appear in bone marrow. See, if we collect some bone marrow and examine it under a microscope, the first erythrocyte which we see is the early normoblast. Okay, it appears first in the bone marrow examination. Okay, early normoblast is the first to appear in bone marrow. Now, the cytoplasm is scanty and deep basophilic. Okay, cytoplasm is scanty and deep basophilic. Now, dark violet, this is the important point to remember. Okay, see, dark violet heterochromatin clumps are present in the nucleus. These heterochromatin clumps are present in the nucleus, which gives the wheel spoke appearance. Okay, see, I'll just show in a diagram, show you the diagram. Suppose this is a a cell which has a nucleus, and there are different heterochromatin clumps like this. Okay, these heterochromatic clumps are joined by a linear sequence, like linear strands, like this. Now, this due to this joining of these heterochromatic clumps by these linear strands, there will be a wheel spoke appearance. Okay, so wheel spoke appearance will be there in which in early normoblast. So there will be wheel spoke appearance due to the connections of clumps by linear strands. See, there are different clumps like this. If all clumps are together joined by a linear strand, ultimately it gives a wheel spoke appearance, na? So in early normoblast, there will be a wheel spoke appearance, And the most important point which we need to remember here is HB appears first time in the cells. Okay, hemoglobin appears first time in the cells. Okay, now we'll go to the next stage, that is intermediate normoblast. Intermediate normoblast. This intermediate normoblast is also known as polychromatic normoblast. Why polychromatic? Poly means many, chromatic means colors. Polychromatic, that is multiple colors. That is why it is named as polychromatic, is the cytoplasm in this stage is chained acidophilic as well as basophilic. Okay, the cytoplasm which is present at this particular stage is chained both acidophilic as well as basophilic, so it is termed as the polychromatic normoblast. Okay, now the what is the main characteristic feature? The nucleus becomes condensed, that is the nucleus shrinks, that is the size of the nucleus decreases. Initially the size of nucleus was like this, now the size of nucleus becomes like this because of the condensation. Okay, now see, I have written here chaining is both acidophilic and basophilic. Okay, now what we have discussed about early normoblast, there will be the wheel spoke appearance, right? There is a wheel spoke appearance, but in intermediate normoblast, there is a checkerboard pattern. Checkerboard. I mean, checkerboard or checkboard, like checkboard. We know that checkboard like this, na? So this checkerboard or checkboard pattern or the uh, heterochromatic clumps. Okay, heterochromatic clumps are wheel spoke appearance in the early normoblast, whereas checkboard appearance in the polychromatic normoblast. Please understand the difference. It is very important for the MCQ point of view. Okay, now HB synthesis occurs and HB synthesis increases. See, here in exam, if an MCQ appears like this, in which of the following stages of erythropoiesis HB is synthesized? In options we have early normoblast and intermediate normoblast, both are there in options. Then what will you choose? Which option will you choose? You need to choose intermediate normoblast. Okay, because HB synthesis increases here. Here it just appears, but here it synthesizes and increases. So in the option, if there is early normoblast and intermediate normoblast, we should choose for intermediate normoblast only. Okay, even if in viva, if any professor asks you in which stage the HB synthesis occurs, your answer should be the intermediate normoblast only. But we need to also remember that the appearance takes place in the early normoblast. Okay, now we'll be going on to the second stage, that is the late normoblast. Okay, in the late normoblast, there is an appearance of pycnotic and small nucleus. What is the meaning of this pycnotic? Pycnotic is simply nothing but shrinked. Okay, condensed, shrinked. All these type of things are called as pycnotic. So pycnotic nucleus and small nucleus, and here we have cartwheel appearance. Cartwheel appearance. Okay, now the HB synthesis is almost completed at this stage, and the cytoplasm is acidophilic. So what can we gain from here? The first acidophilic cytoplasm is appeared in the late normoblast, right? Acidophilic cytoplasm. Okay, so which type of appearance? Cartwheel appearance. Wheel spoke appearance. See, wheel spoke appearance in which one? Come on, comment. So wheel spoke appearance is seen in the early normoblast, whereas the checkboard is seen in the intermediate normoblast. Okay, and in the late normoblast, it is the cartwheel appearance. So cartwheel appearance in the late normoblast. Okay, wheel spoke in the early normoblast, checkboard appearance in the intermediate normoblast, cartwheel appearance in the late normoblast. Okay. Now, we'll go on to the reticulocyte. The next stage is the reticulocyte. Okay, in reticulocyte, nucleus is absent. See, the nucleus size of decreases, and slowly at each stage, the size of nucleus is decreases, and completely it disappears in the reticulocyte. Okay, at the reticulocyte stage, nucleus is absent, and It is slightly larger than RBC. Okay, and remnants of the important point here is the remnants of RNA appears in the form of reticulum in the cytoplasm, hence called reticulocytes. Okay, why it is termed as reticulocytes? Because see, suppose this is a cell, there are some remnants of RNA like this, which gives an appearance of reticulum. Okay, so this type of appearance of reticulum gives the name reticulocyte. Okay, now the last stage, that is erythrocyte. Okay, so this is the final mature cell of erythropoiesis. Okay, now it is biconcave in shape and reticulum disappears and nucleus is absent. Okay, nucleus is absent in erythrocytes, reticulum is not present, it is biconcave in shape. Okay, so this is a matured erythrocyte which is released into the circulation. Okay, I hope you are all clear with the stages. If you have any doubts, please comment. Okay, now we have till now we have studied the process and the stages and their characteristic features of the erythropoiesis. Now we'll see how this erythropoiesis is regulated. Okay, how this erythropoiesis takes place in our body, what are the stimulating factors, what are the maturation factors of this erythropoiesis. Okay, so regulation of erythropoiesis. So this regulation of erythropoiesis can be studied under three main headings: stimulating factors, maturation factors, and factors for hemoglobin synthesis. Okay, so what are the various stimulating factors? The first stimulating factor is hypoxia, erythropoietin, thyroxine hormone, or hematopoietic growth factors, or certain vitamins. These all are the stimulating factors. We'll be discussing each stimulating factor in detail. Uh, okay, so what are the stimulating factors? Hypoxia, erythropoietin, thyroxine, the growth factors, the vitamins. Okay, now what are the maturation factors? Maturation factors are vitamin B12, intrinsic factor, and folic acid. These three are the maturation factors. Now, what are the factors necessary for hemoglobin synthesis? We all know that the RBC cannot survive with hemoglobin. Even hemoglobin cannot survive with RBCs, right? So, when we are discussing about the RBCs, we should also talk about the hemoglobin. Okay, so what are the factors for HB synthesis? They are proteins and amino acids, iron, copper, cobalt, nickel, and various vitamins. We'll study each factor detailedly. Okay, the first and the most important factor is the hypoxia. Now, what is this hypoxia? This hypoxia is nothing but the decreased O2 supply to tissues. Okay, when there is a decreased O2 supply to tissues, hypoxia occurs. We will be studying hypoxia in detail in respiratory physiology. Okay, in respiratory physiology, we will be studying hypoxia in detail. But till now, understand that the hypoxia means there is a decreased supply of oxygen to the tissues. Okay, so when there is hypoxia, there is decreased tissue oxygenation, and when there is a decreased tissue oxygenation, that is when there is decreased O2 supply of tissues, kidneys get stimulated and produce erythropoietin. Okay, erythropoietin. Kidneys produce erythropoietin. What is this erythropoietin? It is a hormone basically synthesized by kidneys. Okay, when there is a decreased tissue oxygenation, kidneys get stimulated and produce erythropoietin, and synthesis of RBC occurs. Okay, so first main factor is hypoxia. The second factor is the erythropoietin itself, the hormone erythropoietin. Now look, the hormone erythropoietin is secreted by peritubular capillaries of kidney. Okay, erythropoietin is mainly secreted by peritubular capillaries of kidney. You will be studying this in the renal physiology. Okay, so peritubular capillaries synthesize the hormone erythropoietin. Okay, and the small quantities are also secreted by liver and brain. Okay, in small quantities are synthesized by liver and brain. So how does this erythropoietin acts and stimulates the erythropoiesis is it enhances or it stimulates the production of pro-erythroblasts from the hematopoietic stem cells. We have discussed, no, in the bone we have bone marrow. In bone marrow we have hematopoietic stem cells. Hematopoietic stem cells convert into uncommitted cells and then committed. and then proerythroblasts appear. So this proerythroblast synthesized from these hematopoietic stem cells by the action of erythropoietin. Okay, now this is the first point, and the second point is development of proerythroblast into mature RBCs. You know that the proerythroblasts are converted into mature RBCs. Okay, in this process also erythropoietin helps. Next, release of mature erythrocytes into blood. Okay, after the synthesis of mature erythrocytes, there must be release of them into the circulation. So this is also stimulated by hormone erythropoietin. Okay, I hope you are clear with this. The next hormone, this is also a hormone, thyroxine. Thyroxine is also an hormone. Okay. Now, how does this thyroxine hormone? How does this thyroxine hormones help stimulating the erythropoiesis? Is this thyroxine hormone is released by thyroid gland basically. Okay. This thyroid gland, no, it releases hormones. These hormones, the thyroid hormones, they increase the metabolic rate of our body. Okay, they basically increases the basal metabolic rate. So this thyroxine also stimulates erythropoiesis by increasing tissue oxygen demand. How it is related to tissue oxygen demand? See, if there is an increased BMR, there will be increased O2 demand also. Okay, so if there is an increased O2 demand, so there will be increased erythropoiesis. Okay, right? Now we have different thyroid disorders also. No, I hope you are familiar with this, right? Hyperthyroidism, hyperthyroidism, in which there is increased secretions of thyroid gland. So what happens here? If there is increased secretion of thyroid gland, there will be increased BMR. If there is increased BMR, there will be increased O2 demand, which increases the erythropoiesis and leads to a condition called as the polycythemia. Okay. So, in hyperthyroidism, polycythemia is common. I hope you are clear. And the next factor, which is most important factor, is the hematopoietic growth factor. Hematopoietic growth factors. These hematopoietic growth factors are interleukins, basically. Okay, these hematopoietic growth factors are interleukins and stem cell factors also. These interleukins and stem cell factors they increases the erythropoiesis. Okay, here are some examples given. Interleukin 3 is secreted by T cells. Interleukin 6 is secreted by T cells. Interleukin 11 is secreted by osteoblasts. Okay, I hope you are clear. These interleukins and stem cells, the stem cell factors, also help in the synthesis of RBCs. Now, here's an important note. It is mostly asked in Viva. This note: the RBC count in males is higher than in females. Why? Because RBC count in males is higher due to testosterone. We all know that the males have testosterone, okay, whereas females has estrogen, okay. So this testosterone it increases the erythropoiesis, okay. The testosterone it increases erythropoiesis, whereas the estrogen it suppresses erythropoiesis. Okay, so this is the main reason males have higher RBC because of testosterone only. Okay. Now, we'll be discussing about maturation factors and factors for HB synthesis in together. Okay. So, what are the maturation factors and factors of HB synthesis? I have written in a tabular format here. Okay. Vitamin B12. It is essential for DNA synthesis in normoblasts. See, we have to remember this table. There is no any other alternative. On repeated revisions, we'll be remembering this. Okay, and I want you guys to present in the same format in the exam. See, in this tabular format, in the flowchart format. If you represent like this, no, there is a high chance of gaining more marks. Okay, I'll be sharing the notes with you guys also. Please try to represent in this manner only. Okay, now what the factor vitamin B12. Okay, this vitamin B12 it increases DNA synthesis in normoblasts and it requires intrinsic factor for its absorption. Vitamin B12 is absorbed no into our body. It requires intrinsic factor. Now, this intrinsic factor is released by parietal cells of stomach. You'll be studying this in the gastric physiology, that is the GAT physiology. Okay, so this vitamin B12 it requires the intrinsic factor for its absorption. Okay, and vitamin B12 also promotes normal nuclear maturation and cell division. Okay, basically it is required for DNA synthesis, cell division. In short, I'll just write in short, this is required for DNA synthesis, maturation. Please ignore my handwriting, guys. And cell division. Okay, next, the folic acid. It is required for DNA and RNA formation, rapidly, rapidly dividing cells in the bone marrow. Okay, see, in this bone marrow we have hematopoietic stem cells, right? These cells divide rapidly, so it requires DNA and RNA. So this is stimulated by folic acid. Okay, this DNA and RNA are synthesized by folic acid, which helps in rapid growth of the cells in bone marrow. See, all these all are almost similar. Okay. Next, iron. Iron, we all know that the HB contains iron, right? We are studying from our 10th class. Okay. So, iron is the central atom of hemoglobin, basically, and it also carries oxygen, right? So, iron carries oxygen, and it is also a central atom of hemoglobin. Next, copper. This copper is required for the absorption of iron. There is no such rocket science in this. So this copper is required for the absorption of iron. And vitamin C. In vitamin C, it promotes general tissue growth and metabolism. Okay, it promotes tissue growth and metabolism, increasing the erythropoiesis, and it also increases the absorption of iron. Okay, now what is the role of these proteins and amino acids? See, we all know that the hemoglobin, right? Hemoglobin, as the name suggests, it contains heme part and globin part. This heme part is from the iron, whereas this globin part is from the proteins and amino acids. Okay, so proteins and amino acids help in the synthesis of the globin part of hemoglobin. I hope you all guys understood the topic. If you have any doubts, please do comment. I'll be replying to it. Thank you. Thank you once again, and see you. Bye.